There are three things that are really interesting. Number one is the use of toll-like receptor 4 inhibitors along with opioids, basically mitigating the side effects of opiates. And then also exercise as a pain management, pain preventative, actually. And some really cool stuff that involves recombinant gene therapy to treat chronic pain.
You're listening to Barbell Logic, brought to you by Barbell Logic Online Coaching, where each week we take a systematic walk through strength training and the refining power of voluntary hardship. Hey, welcome to the Barbell Logic Podcast. This is Matt Reynolds. I'm here with Jayson Ball. My podcast partner, Scott, is actually not here yet. He's running a little bit late, but you're a PhD student, so you're on a schedule, man.
On a schedule, yeah.
So,
I've got about an hour, and then we're going to go hit the microscope.
Perfect. So you've been a coach for us for quite a while. You are at, is it University of Colorado Boulder? Is that what it's called?
Yeah, yes, sir.
And so you're in Boulder. You've been there. You just completed your first year of PhD studies, trying to earn your PhD.
I'm in my first year. So I just finished
your first semester. That's right. It's Christmas. Yeah. It's Christmas. And you are focused on pain neuroscience. Is that correct and fair?
That is correct and fair, sir.
I had you out to the house. It's probably been close to a year ago now. And
yeah, a little over a year.
Super interesting discussion about pain and the science behind pain and what you guys are doing there in Boulder. And so I just wanted to have you on the show and talk to you a little bit about what you're discovering and what it means for us. And we can both sort of explore the theoretical as well as the practical behind what's going on. And so thanks for being on the show, man.
Yeah, happy to be here.
So let's start with how did you get involved in this? Like you've been a strength coach for a long time, actually. You've been doing this for quite a while. And how did you decide to pursue the pain neuroscience side of things?
So I started off being more interested in philosophy. And I was more interested in psychology, kind of theory of mind, and religion and mysticism. And then I got into cognitive science by pulling that thread, and then neuroscience by pulling the cognitive science thread. And when I was at a point with where the gym was ready to be wrapped up in upstate New York with Noah, and we sold that, I moved to Boulder and started volunteering in one of the labs, which was a pain neuroscience lab. And I came to fall in love with the model. So like we do animal research and we're modeling chronic pain, and the fact that it seemed like we could actually get a lot done. It seemed like I lucked into something that was pretty quote unquote straightforward. It's not as hard as trying to say measure something like depression or socioeconomic status, things like that. If you're really trying to like study those things that are complex in people, it's hard to model those in, say, a rat. Whereas pain is more straightforward. It won't translate perfectly, obviously, because we're different. But the nervous system, the sensory systems are pretty similar. That's why like the visual system in primates was able to be studied without hacking people up.
Yeah. And then I remember talking to you about this about a year ago. We've talked about this on the podcast before, that the opiate epidemic is rampant in probably everywhere, but certainly in the United States and the Western world. And you all there at the lab are trying to make some progress in being able to help people deal with pain, but in a way that doesn't get them addicted to opiates. Is that fair?
That's a really good way to put it. Yeah, and if you want to talk about opiates, like that was on the agenda for today.
Yeah, let's talk about them.
So maybe we could start with just a little bit of history for people.
Yeah.
So the opiates have been around for a long time. Opium, there's historical use from 5,000 years ago in lower Mesopotamia. And it wasn't until 1803, I believe, that that it was actually pharmacologically understood. So opium is a cocktail drug, and it's got morphine, which is the principal ingredient, codeine, and another lesser important one that was synthesized into oxycodone. But it's a big cocktail of stuff. So you go and you try to figure out what this actually is, and it's got a bunch of different ingredients in it.
Interesting. I actually didn't know that. I thought that all of those sort of opiate drugs, from heroin and Dilaudid all the way down to, I don't know, tramadol or whatever, is like sort of the most, the least strong, were just really on a spectrum, and they were the same drug. But you're saying that they're actually separate drugs in opium themselves, and that I assume the body then has a little bit different response to each one of those.
So each one of those drugs is going to have a similar protein structure that will bind to an opiate receptor, and they'll have different secondary structures that determine the binding affinity or secondary things like that. Sure.
And so continue with the history. So we get to 1803. Sure. It's sort of understood pharmacologically. We also know, I think most people kind of understand the problem that opium became in the late 1800s, early 1900s, when it was still essentially like you could just go down to your doctor and get a bottle of opium, right? Or
get a bottle of that cure-all.
Yeah, that's right.
Just go over to your boy's house and get a bottle of opium. So it's been known as a problem for a long time. So wherever you find a use of opium historically, you're going to find both its knowledge of its use as a medicine and awareness of potential social harms from it. One of the most interesting parts of the opioid history was the opioid wars. Have you heard about this? Mm-hmm. So absolutely insane. 1800s, where the British government had a state monopoly on opium, and they were at war with China to keep pushing it into China. It absolutely blew my mind, because this is obviously before World War II.
Sure.
And before there was the kind of sovereignty of the state was more or less respected.
So England has a monopoly on it, and they're importing it into China. Is that correct?
Yeah. So basically they're like, hey, China, we want to sell this opium, and you guys have a great big market. Obviously. And then the Chinese government says, actually, we don't like the way this is working in our society, and we don't want to have anymore. So it's outlawed.
Right.
And then, in order to keep selling it, the Navy, the British government Navy, just escorts their trafficking ships with
into China.
Ships. They're just like, no, we are going to sell this. Fuck you.
Yeah. Wow. Most of that was where are most of the opium, the poppy fields? Are they in like Afghanistan, and like where was it being manufactured?
Yeah, correct. That. I think Northern Africa as well.
Okay.
Like that kind of climate.
Okay. So China recognizes there's a problem in the 1800s. Walk me through today, where we've got this massive opium epidemic.
Sure. So when you have something like morphine, and you have the emergence of pharmaceutical companies, you've got Bayer. They're the ones who made aspirin.
Yep.
So they made this wonderful drug in 1898 as a non-addictive alternative to morphine.
Okay.
You want to guess what that drug was?
I don't know. Is it also an opiate?
It's heroin.
Oh, it's heroin. Good.
So they're saying, hey, we got this non-addictive alternative, and isn't this great?
Interesting. So we got this great drug that is far less addictive than opium. It's called heroin, everybody.
Oh, it's awesome.
That's why it's called heroin. It's going to save your life. It's going to come in and save you from the rigors of opium addiction.
Yeah, exactly. So then you have a heroin problem.
Right.
And then it becomes illegal. And then you have a black market.
Mm.
So, fast forward through some fun creation of a black market and an opiate epidemic by what is now considered overprescription.
Sure.
And these days, the cost of opioid abuse is pretty high. So
sure.
one of the review papers I was reading for studying this was on the human side of things, because when you're hanging out in your microscope and in the wet lab, it's easy to lose track of the larger social context of what we're doing.
Sure.
So it's important to to go back and actually understand what's going on in history with this stuff. But something like in the ballpark of 50 billion lost dollars just to the U.S. economy, somewhere around per year.
That's incredible.
And then somewhere around 10 to 15,000 deaths per year, and then a big burden on medical insurance as well. So if you're on a private insurance, if you are an opioid abuse patient, like the average cost that you incur in healthcare is much higher. It's about 8 times as high, not just for treating opiate stuff, but directly opiate problems, but also for comorbidities that come with that kind of problem.
So when you got to the lab, how long had this lab been in operation before you got there with a focus on pain neuroscience?
A long time. So
decades?
Couple decades, actually. So the lab I'm in is the directors actually founded the neuroscience program here at University of Colorado Boulder back in the day. And this lab has 2 sides. There's the stress side and the pain side. And they're both united in that we look at immunological mechanisms for those things, in addition to neurological mechanisms. But that was the kind of big breakthrough and claim to fame that's kind of come out of this lab.
So is the focus at this point in 2020 really to help really change or really put an end to the opioid epidemic long term, to be able to treat pain in a way that is actually healthy and tolerable and has far less side effects? I mean, what's the ultimate goal at this point? While I understand it's never going to be, hey, let's leave the original foundational idea of let's continue to add leaves to the tree of knowledge. We want to continue to learn more about this. And we've talked about this before on the podcast, and it's everywhere. We know very little about pain, right? Like, pain is just people, and when people have pain, they want an answer to what is causing the pain. And for years, our answer has been like, we don't really know sometimes, right? Like, and when you go to a doctor, it puts the onus on the doctor or the MRI tech or the physical therapist or whatever to read the thing to tell you, like, you want an answer, they're paid to give you an answer, and so they're like, you've got a herniated disc, a grade 1 herniated disc, L5S1, whatever, right? And yet we know that there are lots of people with grade 3 herniated discs that have no pain symptoms whatsoever. We know there are people who experience tremendous amounts of pain symptoms with no cause that we can find, right? At least not a physiological cause. And so what does this look like in 2020? What's the direction that you guys are going now in the present and pushing towards the future?
So one of the things you were just talking about, which was really good, is the idea that if you go in and you look at an MRI, that the assumption is that's what's creating a pain state. And they might be correlated. I haven't looked at the human data very much. My understanding is they're not correlated that well.
Sure.
But the thing to remember is that, like, pain is an output of the nervous system. It's this kind of motivational, emotional side of this downstream motivational, emotional system of nociception. And if you go in and look at, say, an X-ray or an MRI, you're looking at this gross anatomy. That resolution is not. It might not be fine enough to tell you about what's happening in the nervous system.
Sure. I mean, there is no nervous system on a X-ray or an MRI, right? Like, there is nothing to see from a nervous system perspective.
You can see the spinal cord.
Right.
You can see this big old rope.
Sure. But outside of that, that's it.
You can't quantify, like, what cells are sending what signal.
Sure.
And that's the problem. And I think that it's easy to see that and say, well, look, the X-ray and MRI data doesn't correlate to what the patient reality is in medicine. Then, therefore, it's not a physiological thing. But I would say, like, just kind of a first principles thing is that any subjective experience you can have is a physiological thing, because a subjective experience is based in the substrate of the neural system.
Sure.
But if you're looking at it from, if the thing we care about is, say, there's a population of neurons that are overactive, and that's being interpreted upstream as in some catastrophic way, then you might not be able to tell that with an fMRI or an X-ray or something like that. So
Sure. Yeah, this is the problem with really pain diseases that are like a fibromyalgia, right? Where we can't find the source of the thing, but like we recognize that the person clearly feels pain. Like we're not saying, so when there's been a lot of discussion about these pain type diseases that are psychosomatic, and maybe that's true, maybe it's not, but what we know is that like they're actually, like they're not faking the pain, right? Like we believe that the person feels pain, but figuring out the source of that pain or why that pain is occurring or why the nervous system is sending these signals, like this hurts, we don't exactly understand that yet.
Yeah, and what we're studying here in terms of like the neuroscience of pain is in rats.
Right.
So anything we get here has to be cashed out in medicine down the line.
Yep.
So even if we say, hey, we think we've got a pretty good understanding of how this works in a rat, that doesn't necessarily mean that it's going to be the same in a person.
Sure.
But you use a rat as a model because you can do more precise controlled studies on it than you can with people.
Sure. Like, and I mean, realistically, you can just do potentially more dangerous things. You can test things that are, while there are people who I'm sure have some issue with you testing on rats, obviously, there are very few people on Earth that would say, well, we would rather you test it on humans. Like at some point you go like, look, if we're going to solve this problem, we got to test it on something, right?
Yes, sir. Yeah.
And
that's
so
that's a good way to put it.
Over the last, so you've been there for a few years, you're one semester in on your PhD, but you've actually been in the lab for a while now. What are some of the more exciting things that you're seeing coming out of the lab that you guys have been testing for the last several years that you're like, this is super wild, like super interesting?
Okay. So there are three things that are really interesting. Number one is the use of toll-like receptor 4 inhibitors along with opioids. So basically mitigating the side effects of the side effect profile of opiates in terms of making pain worse. Because one of the things that was surprising is that if you use pain in a neuropathic model, the pain gets worse. Or sorry, if you use opioids in a neuropathic model,
right,
the pain gets worse after you stop using it, and that's beyond just the withdrawal stage.
Sure.
And then that mechanism was discovered in 2008, I believe, by Mark Hutchinson as being immunologically mediated. And then the follow-up to that has been pairing opioids with something that blocks that signaling cascade, that off-target signaling cascade. And so getting a better profile for opioid use is the one of the themes. And then also exercise. We also study exercise, which is really cool. And as a pain preventative, actually. Mm-hmm. And some really cool stuff that involves recombinant gene therapy to treat chronic pain. So those are the three main threads. You got exercise, better use of opioids, and kind of
and the
next
recombinant
generation
gene therapy.
Tech.
Okay, so can you talk in layman's terms, like what are the things you're doing to pair with the opiate usage to negate some of these negative side effects? And is it specifically focused on, I assume it's focused on both while the person or rat, in your case, is taking the opiates, but also post after they get off, right? You said there's this massive like problem of pain increases when they get off the pain meds. Like now my back still hurts.
Yeah, so I'll back up and give you some context. So the basic phenomenon here is that when the rat would have ongoing chronic pain and was then treated with a 5-day course of morphine.
Hold on, back up, because you and I talked about this earlier. How do you know the rat has chronic pain?
So the measurements, there are a few measurements you can do. The first one is for allodynia, and allodynia just means treating touch as pain. So if you're a chronic pain patient, there's a good chance you've got some allodynia going on. That's where your clothing hurts, for example. If your partner touches you, it's felt as pain. It's where your nervous system is making the pathological wrong assumption that touch is nociception.
That sounds sort of crazy, but the reality is I think almost everybody's actually experienced this before. For me, like when I've had the flu, right? And you talk about like
Yes, sir.
the body aches of the flu, and you can't quite explain it, but
Absolutely.
like everything hurts. I'm laying on my bed. Anywhere that I'm laying on the bed hurts. Like my clothes hurt on me. And it's very rare for me, but if you've ever been really sick and you're just trying to explain, somebody's like, what's wrong with you? Like, man, it's everything hurts. I'm just really achy. I can't quite explain it. Like, that's it, right? That's what we're talking about.
Bingo.
Yeah. Okay.
Bingo. And that's we can come back to that if you want to, but that's that's like a perfect example because it's mediated by inflammation. It's the effect of an inflammatory state in your body to fight a pathogen that produces sickness behavior.
So again, though, very practically, like how would you know if a rat has that?
So if you test them, what you do is you put a rat up on a little rack so it can just walk around in this little wire rack, and then you have a set of calibrated hairs. So imagine like a guitar string.
Yep.
The guy who did this originally was using horse hairs.
Okay.
But you have like guitar strings of varying thicknesses that will bend when you apply a certain amount of force.
Okay.
You have a baseline for the rats. So say on average for male rat, it takes about 10 grams of force applied to its paw, and it will pull it up and flick it.
Okay.
It's like we as humans, as researchers, we interpret that as an ow reaction,
Sure.
not just a touch. It goes from being something where it's like, well, what are you doing? Or I don't even feel this, to it jerks its leg back. Yeah. So this is, and then you have a set so you can see where the rat is, where its pain threshold is, where its withdrawal threshold is, we'll say. And then you can chart over the course of a model, of an injury model, of a pain model, how sensitive it is. So it goes
wild.
from, say...
And there's nothing that you're doing to the rats ahead of time. Like you're just literally sort of recognizing or identifying that some of these rats have rat fibromyalgia. I mean, it's like, okay, so it doesn't take 10 grams. It takes only 5 grams for them to pull back and go, this hurts.
Yeah. So the basic idea of how you're going to do animal research is you use a model like this. So there are several popular pain models. This is one of them. And then you profile what's happening, and you make hypotheses about how either the injury itself could be resolved faster, or the pain state could be mitigated. And you apply that intervention to the cohort and see if their outcomes differ along behavioral measurement.
Okay.
And so you can do all kinds. The one I told you about is called Von Frey test, where you poke the feet.
Yep.
That's like the most basic kind of allodynia test. But you can do all sorts of testing. You could do it where you put them, you expose them to like a hot floor, where a normal rat wouldn't care. It's like, if you get a sunburn,
sure,
the hot shower feels like it's burning.
Yep. So
it's the same kind of thing. So you can recognize like a heat sensitivity.
Okay.
You could do something more cognitive, where they know that there's a reward on the other side of a maze, and you put a prickly surface in between them and the reward, and a non-neuropathic rat will go and get the treat.
Yep.
And a rat that's in pain will say, oh, no, that hurts. I don't want to do that. And it'll just kind of stay. So there are a lot of ways you can assay this, but the basic playbook is you have a model of the phenomenon and a behavioral outcome, and then you're trying to intervene in the nervous system or the adjacent mechanisms to change the end behavioral outcome or the outcome in the tissue. And ideally both together.
Let's talk about then how you intervene. So then what do those interventions look like for the rats to try to, what are the promising things on the horizon there?
We should probably talk about exercise too, but I
definitely want to get there for sure.
The cool stuff, it depends on where you want to look in the model. So if we're doing a morphine model, the really cool thing is that you can prevent the negative effects of morphine. This is setting aside things like addiction, because we're not playing with the dopamine system. That's a separate problem for people to solve. And actually, I take that back now, because I think that this might actually help with that as well. But the way we're looking at it is just preventing the allodynia that occurs with morphine use. And allodynia was just like the feeling of touch is pain.
Yep.
The basic world here is that after a 5-day course of morphine on a chronic pain rat, the time course for them to resolve back to baseline takes longer, and if you give it to them later down the road, you can produce a dip back into chronic pain after this 5-day course. And the reason this happens is because the first question is like, well, is it opiate receptor signaling? That's the first thing you think of. It's morphine. So maybe this is more mediated by the opiate receptor.
Yeah.
And the answer is no. So you can delete all the genes for opioids. So you can have a mouse line that this has been done. So you have a mouse line where they don't have opiate receptors, and you can still induce those effects.
So they don't feel.
So they don't feel
relief. Right. They don't get any of the pain relief from the opiates. Like they don't feel anything from the opiates, but they do have the pain after effects.
Yep, they get the hyperalgesia, they get the naive tolerance, they get all the stuff that we don't like about morphine.
Okay.
So that's one of the ways you can get pharmacologically closer to what's happening. That's what people have done back in the early 2000s, is use a mouse line where you've deleted all the genes for these receptors, or you can use molecules that are similar to minus morphine, which is the kind of morphine that is used. So morphine's a stereo. They're stereoisomers, so it's like a mirror version.
Yep.
Left hand, right hand. So you can use a stereoisomer of the molecule that doesn't bind to the classical opioid receptor, and you still get these negative effects. You don't get the pain relief, but you get the negative effects. So the question is then, well, what the hell is going on? And what people found out in the late aughts was that the innate immune receptors were being activated. So there's imagine in all of your tissue and nervous system too, you've got immune cells that are surveying the landscape and looking for problems. And what was found was that opioids were binding to a motif in an immune receptor that is scanning for bacterial pathogens. And specifically, it's this thing called MD2, which is an accessory binding protein to toll-like receptor 4. And toll-like receptor 4 is this big, like jungle gym type receptor that looks for evolutionarily conserved signs of damage or infection. And what's happening with opioids is they're, in addition to binding the opioid receptor and providing all the effects that we want, they're also binding to this off-target thing that is actually critically involved in producing and maintaining pain states.
Yeah. So then do you address that specific receptor in your treatment to say, okay, then with the use of opioids, we're gonna make sure that they can't bind to that additional offshoot receptor to cause that cascade of effects. Yep. And how do you do that?
So you'd use an antagonist. That's the most common way to do it. So you'd when you're administering an opioid, for example, you also block the binding site that's associated with toll-like receptor 4, and then you can rescue the pain state. And then one of the cool things about this was that the use of this kind of antagonism, this innate immune system antagonism, is useful not only with opioids, but if you have just straight up chronic pain state without the use of opioids, because the immune cells that are in charge of maintaining the pain circuit are activated. And this is what is looking like is going to be the most fruitful way forward for chronic pain management, is this immune angle. So you're not targeting the exact neural circuit for pain per se, neural circuits for pain per se, but you're trying to target the immune systems that are in charge of maintaining their excitability.
Yeah.
So, and that's something that was, I think it was quite controversial at the time. Everyone was focused on neurons, and then, someone comes along and says, actually, these kind of boring cells that people forgot about, that seemed like they're just, like little statues that hang out and make sure that the glutamate's cleared and out of the synapses, and there's no bacteria around. Those are actually involved in producing and maintaining chronic pain states. Right. And if we target them, we can alleviate the chronic pain. So you remember when at the conference in 2018, I think, when Sullivan and all those guys were talking about hip and knee replacements?
Yep.
So one of the slides they had was the pathology of osteoarthritis in the knee.
Yep.
And one of the points was that they made was this: there's no, aside from exercise, there's no known like pharmacological targeted intervention for this pathology, this tissue pathology, because now you have this feedback loop that's happening in the immune system. It's keeping it inflamed and keeping all the pain fibers there sensitized. So that's the target. And that's what they're actually in human trials right now with the gene therapy to do this.
Yeah, so I was going to say, so really like this sort of bridges into, you talked about the three really exciting things. This is the thing that really applies to all three of those things, right? So if I understand correctly, when we start talking about the first thing that you said was interesting, it's the idea that we've now have identified these receptors that opiates bind to and cause this sort of pain highway, this chronic pain highway, and we can put a stop to that with or without the use of opiates, right?
Yeah.
Meaning that there is a day coming when either A, there's this thing that's added to opiate drugs which make them still, you get the essentially the positive side effects without the negative, or even better, that the day is coming when we don't have to use opiates at all and we can just turn off the pain highway by treating these receptors with these antagonists and say like, no, right? Is that a very layman, dumbed down way of saying
that's a really good summary. The only thing I would add is that it doesn't mean you won't use opiates at all, because it looks like, especially if you're doing something like surgery,
right?
Something very intense and acute. I don't. This is just me making predictions about the future from my teacup here.
Sure.
But I don't think we'll have something that will replace opiates for a long time. They're great for that.
But you're talking about long-term, relatively low dose usage. I've been on oxycodone, Percocet, Tramadol, whatever, for 3 years straight, because if I get off of it, immediately the pain comes back. That's the thing that this addresses.
Yeah, it'll be trying to help exactly that kind of chronic pain management.
Now, moving to the next step, let's get into the exercise, because it's interesting, because in spite of the fact that we've got all of these like super cutting edge technologies of like how to do these things with the receptor, or even further down the line of how to literally play with genes to not, well, I want to get there last. You've got this sort of like archaic caveman thing, which is like, oh, by the way, exercise literally is the best thing that we can do to fix this without drugs or any sort of gene therapy whatsoever, right?
Yep. That's my understanding. And this is something where it'd be great to have someone who's more familiar with the human literature, like the actual medicine side of things. But my understanding of it is that for long-term pain management, the exercise is pretty good. It's the best thing we've got. And there's basically no drug that comes close right now for long-term pain management.
Well, and we can go back to, I don't want to oversimplify this, and obviously it's easy for me to do so because I'm so ignorant about the whole thing, but I've stated for years that I again, go back to the fibromyalgia issue. I know that people who have this really experience pain.
Yes, sir.
But if you go to professional athletes, Olympians, and you can look at any of the other diseases that affect the rest of the human race, like there are professional athletes, Olympic athletes who have asthma, who get cancer, who whatever, but none of them seem to have fibromyalgia, or these, at least the incidence of chronic pain in professional athletes seems to be insanely low. And the thing that they have going for them is that they are super active in doing their sport and exercise all the time. It's the same sort of thing. If you think about just the strength sports, you think about people who compete in strength sports and powerlifting or strength lifting or strongman or whatever, it's pretty rare that somebody's like, boy, I just struggle with chronic pain all the time. Why? Well, like, is it maybe it's correlation but not causation.
Yeah, you'd have to make sure that you did the study the other way, or look for causality the other way, because you could have a kind of survivor bias, so that people who are elite athletes are, have other things going on for them, and if they did have chronic pain, they would have stepped out.
Right. Yeah, it makes sense. Sure.
But in general, that kind of experience, if you're training people, or if you know people who exercise, that they tend to have less pain. that kind of experience, as far as I understand about the medical data, would kind of match it. And then it definitely is backed by the animal research. And so there are a couple of cool things here that are looking into the actual mechanisms. And there are two really interesting models. Number one is after you already have chronic pain, using exercise as an intervention. And number two is, and this is the one that we're studying, is having exercise before an injury,
right?
And the outcomes of chronic pain.
Sure.
So if you let the rat cohort exercise on a wheel, they just get to run as much as they want. They get a wheel and they can run for 6 weeks. Or this couch potato cohort, and then both get injured, and then from the injury on, neither of them gets to run.
Sure.
And you could the thinking was, well, once you have an injury, exercise is good for you. And what the model we're playing around with is, before you even have an injury, exercise is good for you in that you are less likely to develop the pain you develop from a traumatic nerve injury is going to be less intense or less long lasting.
Yeah.
And that's really interesting. And we're actually looking into like the mechanisms that are involved with that.
So you say looking in, but I assume there's some sort of early hopeful findings, or that, I mean, are you seeing that, yeah, that's actually the case? Like the rats that are doing the exercise before they get injured, the injury itself doesn't have such a negative effect on them compared to the ones that are the quote unquote couch potato rats.
Yeah, and that was I think that was first published in 2016 by Peter Grace. Shout out to Peter Grace. And that's why we got the grant, is because that was the original. So he's a
PhD scientist there at your lab. Peter is?
He's at MD Anderson Cancer Research Facility in Texas.
Yep.
But he was here at this lab.
Got it.
And we share that grant with him.
Okay.
So basically the idea was, Peter published this effect where it's 6 weeks of exercise before the injury is less bad.
Yeah.
Or the pain outcomes are less bad.
Yeah.
And then the NIH says, well, we want to know why that is. And he and my advisor made a proposal to say, we're going to look at mechanisms A, B, C, D and publish on those. And so that's what we're doing now. We're looking at basically what's happening at the cellular level to cause this.
Yeah. Yeah, it's interesting too. Like, I don't want to put words in your mouth, because you have to walk a fine line between the fact that you're a scientist and a strength coach, which is interesting. But in my experience, and again, don't want to speak for you, but I think you would agree with me, in the thousands of people that I have strength trained over the years, a very high percentage of them come in with some sort of pain. And within a few weeks or months of strength training correctly, proper form, and consistency, a overwhelming majority reach back out to me and say, pain's gone. Pain's gone away. Like, my back doesn't hurt anymore. That sciatic nerve pain I had running down the back of my hamstring into my calf I don't feel it anymore. And you go, yeah, right? And it's wild, because I see these two like weird worlds collide of like the most sort of like archaic Cro-Magnon humans have been exercising for the entirety of human history. And at the same time, like, you're in this lab doing these cutting edge research things. And it's like, you start to realize, like, actually there's this thing that everybody can do. We can't 100% prove it yet that it works for every single person. We're not saying that. But the experience is, both with our experience in the weight room with humans and with your experience in the lab with rats, that it seems like exercise makes a tremendous positive impact on pain in both before and after the injury.
Yeah, absolutely. If you want to go talk about like gene therapy, like some sci-fi shit.
Yeah, for sure.
Before we do that, I just want to plant a flag for anybody who's interested in the exercise research. So the exercise as an intervention to a pain state, look up Kathleen Sulka.
Okay.
She's got a lot of really cool...
Sulka?
Yes, sir.
Okay.
And she's got a lot of really cool data on the effects of exercise in the descending pain modulatory pathways, so like from the brainstem down, and basically periaqueductal gray the medulla down to the spinal cord, and the effects that it has at the level of signaling there, just exercise.
Yep.
And so basically, it's similar to it's the same circuit that morphine activates for pain relief.
Yeah.
So, and she does a really good job of looking at that like pain relief circuit and the immunological side, and they do really cool causal. They study causal mechanism where they'll take this hypothesis about a certain immune signaling cascade and they'll block it. They'll knock a certain signaling molecule they think is critical out, and they'll compare the differences. And so it's really good science. Shout out to Kathleen. And I actually talked to one of her postdocs at Society for Neuroscience, and they're doing a linear progression strength training model with their rats too. That it sounds really cool.
Yeah, that's super cool.
It's not published yet, but when it does, I'll post it to the group.
Yeah, that's awesome. So when you first came on, you talked about these 3 things that were very interesting in what's been coming out of the lab and the studies that you've been doing. We addressed the first of the connection between opioids and the immunoreceptors, these things like the pain highways, and the relationship between them and how you're working with them. We've talked about the impact that exercise makes on pain. Some of the most interesting conversations I've ever had with you over the last couple of years is this like frontier of what's going on the genetic side and the gene side. So let's just take a few minutes and just like let's have an amusement park ride here and blow our mind a little bit. Like, just what are some of the crazy things that you're seeing?
Point me in whatever direction you feel like is most fun. But
sure.
When I start going on this stuff, this is why it's so damn fun to be
trippy.
A scientist,
sure.
A neuroscientist specifically. Being a grad student is just great.
So what's on the horizon there?
So this is not the things we're playing around with. To be clear, they aren't CRISPR.
Sure.
So everybody's heard about CRISPR, right?
Yeah. If you haven't, go to the TED Talk. It's actually CRISPR. Is that right?
Yeah.
So go check out the TED Talk on CRISPR if you haven't checked out CRISPR. Super interesting. So yeah, we're not talking about CRISPR.
So CRISPR is where they go in and chop genes out and put new genes in,
right?
To like a chromosome. They're editing the gene of the cell that already exists.
Yep.
One of the gene therapies we get to work with, and other ones that are on the horizon and like in human trials now. You can look, if you like go to PubMed or Google Scholar and you look for gene therapy human trial review, you'll be able to see a list of 100 or so. I think in 2019 they were under human trials. So these are little constructs that are plasmids. And quick review for people who haven't in biology in the last 20 years, the plasmid is just a ring of DNA. That's the best way to think of it.
Okay.
So if you have a animal cell, it's got these funky looking chromosome things where these tightly dense, highly regulated DNA protein structures. But if you go into a bacteria like E. coli, the DNA is in this kind of loose ring. It's accessible, and the bacteria are swapping genes in the form of small little plasmids, and that's just a circle of DNA. And what you can do with these is put in genes that you want and put in regulatory sequences so that you can change when they're expressed under what circumstances, so that the cells that take them up can express that genetic material under conditions that you specify.
Right. So it's genetic. If this, then this.
That's a perfect way to think of it.
Right. Okay.
So in this whole immune-mediated pain, chronic pain model, so this paradigm that we're talking about, where chronic pain is highly mediated by the glial cells that are in charge of maintaining the excitability of pain systems. One of the things you can do to them is hit them with nature's own anti-inflammatory. This what's called interleukin 10. It's a cytokine. A cytokine is just a small molecule that is used for immune signaling.
Yep.
That's what it means.
Yep.
So the same thing when you have the flu and you get really sore and you get a fever and you don't feel like going out and doing stuff and you just want to stay in bed and shiver. All that stuff is mediated by a cascade of interleukin 1.
Okay.
And that changes your nervous system to change your behavior, produces a fever, et cetera, et cetera. That's a cytokine. Interleukin 10 is a cytokine that basically tells the nervous system to go back down, the immune system to go back down from an excited state, where it's facilitating, among other things, nociception and pain signaling.
Okay.
So in the presence of this cytokine, it inhibits the release of things like interleukin-1. It blocks transcription factors that transcribe that gene. It reduces the amount of antigen that your immune cells are showing to your immune system to get it rolling. It's instead of something like, say, a prostaglandin inhibitor, where you have this kind of local, like an NSAID, ibuprofen or something like that. Where you have this kind of local blockage of some minor class of inflammatory mediators, just like swelling and stuff like that. This is something that goes to the immune cell phenotype and shifts it back to its quiescent state.
For the entire system?
So wherever you inject it.
Okay, so it is
what's
specific
in
to
human trials right now is in knee joints.
Okay, got it.
And that's nice because you've got this environment that's kind of isolated. So you wouldn't want to do like this. You would have to be very careful if you're doing something that downregulates the immune system. Like that, you have to be careful with that kind of stuff.
Sure.
Make sure if you have a situation where you have an immune dysregulation that's mediating a pathology, this could be an appropriate tool. But if you just have, but you wouldn't want to just spray it everywhere.
Sure. Right.
Put it in the water.
Sense. Right. So it needs to not be systemic. It literally needs to be acute to specific areas that are experiencing pain.
That's the only way we've used it. Yeah.
Right.
And I couldn't see any reason to do it systemically.
Any other way. Right. So explain what does that look like for somebody who's got chronic pain in the knee? And what if they have chronic pain in the knee because they have a torn ACL or a torn MCL or
So here's the thing. So this is something where we're blind to the human data now. And my PI, the investigator, has been working on this particular gene therapy for something like 10 years. And it's in human trials now, which is great. But we're blind to the data. All we know is, hey, things are looking good enough that we're going to keep going. That's just what the FDA tells her.
Okay, I was going to say why. Like why blind? What are they trying to protect?
When you have a vested interest in something, so you've been working on something for 10 years, your career's
want it to come out positive.
This is your blood, sweat, and tears.
Sure.
And then you need to give it to a third party to validate it.
Right.
That it actually works. So they do double-blinded. They aren't involved with you. It's somebody who's impartial. Sure. And their job is basically just to shut down everyone's hopes and dreams and make sure that that no bullshit gets by.
Right.
But let's talk about it in there. You can actually, if you Google Linda Watkins dog, you can see some video footage of this gene therapy in dogs.
Okay.
There's a local vet where they have she has a collaboration with, where he gets this it's kind of like a compassionate exemption, where basically if the dog is in such bad chronic pain that the owners are going to put them down, then he says, we can try this thing now. It's not approved by like the doggy FDA,
sure,
but it's effectively a compassionate exemption.
Right.
It's, what do you have to lose? Your dog was going to die, or you can try this thing.
Correct. And
you can see like before and after use with that. But the basic thing you want to know with this is this treats the underlying immune phenotype. Now, if you have something that is continually irritating the area, or you have, say, your ACL's torn in half, won't heal that.
Right.
If you have a piece of metal in there that's sawing a hole in some of your tissue, it won't stop that.
But will it turn the pain receptor off for that enough to be able to be like, there is this injury, but I still don't really feel the pain?
Well, it depends. If it's causing an ongoing injury,
okay, got
like
it.
Every time you take a step, it's sawing a hole in your bones.
Right.
Then you're going to get more signaling, more of an injury state.
Sure.
But if you have something that is where the physical pathology is, for all intents and purposes,
normal.
Well, with the exception of, say, the neuroimmune signaling,
sure,
where the actual tissue damage has been resolved,
right,
but you still have this chronic signaling, this activation of that area,
sure,
then it'll address that phenotype. It addresses the signal that that area is sending back to the central nervous system.
So theoretically, even a post injury it's post injury, post op, it's been repaired, it should be fine, but the person is still experiencing pain in the injury, even though there's no injury apparent anymore. This would be the kind of way.
That's exactly how I would think of this being used.
Yeah. Okay.
Among other ways, we're trying to, because the human trials are in knees, osteoarthritis in the knee.
Yep.
And what we're trying to do right now is find ways to get it into the lumbar spinal cord. So we're trying to get it in the central nervous system, and getting it in the brain itself would be probably last, because you have to like inject into the brain, which is not what you want to do if you can avoid it. But what we're trying to do is do something where it's basically a non-invasive situation where you do like a spinal tap, put it in the cerebral spinal fluid, and have it diffuse into the lumbar, for example. So we want to try we're trying to work on delivery methods for central nervous system stuff, but right now it's just in periphery, and that'll be the first use case.
Sure. And so I don't want to fortune tell here, but I can see that what the potential is that this is potentially the future replacement of opiates. It is
for long-term pain management.
For long-term chronic pain management, correct?
Yeah.
Right. That's the goal. That's the goal, at least, right?
And the like, to be clear, this is just starting to get into humans, but the last 20 years of animal research has revealed that these immune players, because visualize this. You have a neural synapse, neuron A talking to neuron B, and they're exchanging chemicals, depolarizing, sending signals. And that's kind of the way it works from, your fingertips to your brain. Now, that synapse is not as simple as just a neuron and a neuron swapping notes. There's a third player, an astrocyte, that's in charge of how much signal is going to be in there for how long. And there's the microglia, that are in charge of the local inflammatory environment, that are changing the excitability of the neurons. And the last 20 years of animal research has revealed that in every clinically relevant model of enhanced pain in animals, so spinal cord injury, bone cancer, multiple sclerosis, chemotherapy, peripheral nerve injury, herniated discs, there's some like rat migraine studies too, that if you hit this immune mechanism, you suppress pain and return it back to normal in every single model.
Interesting.
So this is something, we'll see how well it translates to humans, but it's looking good. And the data we have on at least this one that I'm close to is where they've got, I think Linda did horses as well, but it's like rats, dogs, horses, and now humans. Like, say, rats and dogs. I know the interleukin 10 gene therapy they're using is the human gene. So this is so, fingers crossed it works for people, but like this is kind of this is an evolutionarily conserved system. It's an old part of the innate immune system.
Right.
I'm excited, but I can't fortune tell here.
Sure. But so interesting especially when you look at the opiate epidemic that we have in the Western world. It's like there's hope out there that there is a thing that can. Absolutely. There's work being done in labs, and is the University of Colorado at Boulder sort of the foremost lab that's doing this sort of research? Or are there 5, 6, 7, 12 out there that are doing this type of thing in the U.S.?
So that's a good question. We went to APS this year, 2019, and I'd say it's not 1,000 labs, but it's not 10. It's probably
it's quite a few,
on the order of 100,
okay,
something like that.
And most of those are university-based labs, or some of them are there a bunch of private companies doing this as well? I mean, do you think? So
private companies will probably come later, as far as I know.
So Eli Lilly, Genentech, are they still a company? These companies that were like the pioneers of like gene therapy, they're doing that stuff too in the private sector?
I don't know. I'm not aware. I haven't read any of their stuff if they are.
Okay, got it.
Yeah, it's totally possible.
Sure.
But I'm not aware. The I'll put it this way: investing in new pharmaceutical ideas is not the best investment strategy for your money. If your goal is to make money with your money,
sure,
investing in new radical pharmaceutical stuff is not the best way to do it.
Well, especially if the long-term end is we actually don't even have to give a drug, but rather we just have to adjust a gene, right?
Sure. If you have let me rephrase that. You make more money if you have somebody on a drug for 10 years than getting a single injection
right
once and then maybe getting another one 6 months later and then being done.
Right. Yeah.
So
sense.
it's not I'm not at all going to get on a soapbox and complain about that.
Sure.
That's
just I've actually
never heard you.
I've never heard you complain about that. You're not really. We've never had a conversation where you're like, there's just too much damn money in the opiate industry and they're going to keep us down. And it appears that for the most part, you all have freedom from those dollars to do the research that you need to do to try to make this thing work. And let's say it does work, and I know that's a big if, it's still, what are we, a decade away from it being used on a routine basis in humans, and that's if all goes well?
Probably not that long.
Probably less. 5 years-ish?
That sounds about right.
Okay. I mean, that's a big deal.
Yeah. No, if this kind of, when this kind of research is effectively translated into a human medical management paradigm, it'll be it's a whole new toolkit for medical professionals to take advantage of in their practice.
Yeah, absolutely. That's super interesting. Dude, yeah. This has been super interesting. Thank you for being on the show. So the interesting thing, again, for our listeners, like while we get to geek out on the future of pain and like pain neuroscience and where the studies are going, it's still very interesting to me that our listeners can hear this and be like, hey, the best thing you can do today right now is exercise, is train.
That's correct.
Which is pretty cool. Dude, thank you for being on the show. This is super interesting. I could hear you just talk about pain. Pain is such a wild thing in our culture and the impact it's had really on the strength community and in the culture. Like, who listening to the podcast doesn't have a family member who struggled with opiates at some point in their life? It's just so prevalent here that to say like, hey, there are some things both that we know we can do now and some things on the horizon that are coming that can hopefully set us free from that as a culture is pretty incredible.
Yeah, I totally agree. It's a good field to be in. It's going to be it's important to a lot of people and to society as well to fix this problem. Agreed.
Dude, thank you for the work that you're doing. Thank all your I don't know, professors, mentors, doctors, PhDs that are up there with you. Thanks for being on the show, and we'll have you come back and talk more about this and geek out some more in the future.
Yeah, I'd be happy to, man. It'd be really fun to talk about the psychological side too.
Yeah,
like kind of.
I think it would be really interesting to have you on with Pewter, and we'll talk. We'll have an actual psychiatrist and you, and we can see like the impact. And of course, he's done Dr. Pewter, which is sort of our the local Barbell psychiatrist. He's done a lot of shows. He's got a show, the Psychiatry and Psychotherapy, where he talks a lot about the opiate epidemic in the Western world and the impact that it's had on us. And so it would be interesting to talk through some of the psychological impact that pain has and pain treatment for sure.
Dude, I'd be so down.
Thanks for being on the show, dude.
Thank you.
We'll talk soon.